project is on the relationship between nitrate and nitrite concentrations and E. coli bacteriophage abundance in environmental saltwater conditions. The problem that we addressed in this project was how will the differences in the concentrations of nitrate and nitrite affect the abundance of E. coli bacteriophage in environmental saltwater conditions.
We hypothesized that independently, both the nitrite and nitrate concentrations would be higher when there's more bacteriophages in the water, since E. coli use nitrate as an energy source. And depending on each other, the nitrite to nitrate ratio would be higher when there's more E. coli and thus more bacteriophages, since in the process of using nitrate for energy, the E. coli convert it to nitrite. For the procedure, first we collected 3 different samples from 3 different locations: at the mouth of the canal, in the canal, and in the bay area outside of the canal. We brought those into a lab and let them incubate overnight with some E. coli bacteria to enrich any bacteriophages that each sample might have had. Then we filtered, centrifuged, and extracted the bacteriophage out of it. We then plated the bacteriophage with E. coli and top agarose on a prepared agar plate. We let that incubate overnight at room temperature, and the next day we came back and we counted any plaques that may have occurred in each plate to see how many plaque-forming units or bacteriophages we had in the water samples. We did a positive control and a negative control during the experiment. For the positive control, we added T7 bacteriophage instead of any sample, and that was supposed to clear the plate completely. And for the negative control, we added lab water instead of the sample, and that should have just not had any bacteriophages whatsoever in it. Then we tested our samples for nitrite and nitrate concentrations using a test kit with ampules and a colorimeter.
In order to perform an analysis for our experiment, we also made a calibration curve in which we tested known values of nitrate and nitrite to determine the accuracy for our experiment. For each of our samples, we took 3 from each location, and in L1-1, which was inside the canal, we didn't have any plaques on any of the plates that we plated. In L2, which was at the mouth of the canal, we had 1 sample that had around 8 million plaque-forming units per milliliter, which you can see in this picture here. It has lots of plaques that we had to count. And L3 was in the Bay Area, and that also had 1 sample that had plaque-forming units per milliliter, and that had around 25,000. In this graph, we can see how we plotted nitrite concentrations against the number of bacteriophage plaques, and you can see that way up here is the one that had around 8 million, and the one that had 25,000 is much lower because 8 million compared to 25,000 is very large. And so it does show that the ones that had larger, higher nitrate concentrations did have higher bacteriophage the bacteriophages in the sample, which correlates with our hypothesis. Over here are our calibration curve graphs, and you can see that they both didn't differ by the same amount, so we weren't able to establish a constant in order to correct our data by. And this picture shows the ampules that we used in order to determine the nitrate and nitrite values, and the 3 on this side here, they all measured 0.01, but you can see that they clearly have different colors, so that shows that our measuring device was somewhat off, which resulted in somewhat strange data. Additionally, the nitrate concentrations in our samples were so low that the devices we used to measure it were not able to pick it up, so we weren't able really to use that in our experiment to compare our data.
While our instrumentation did not allow us to measure all that we had hoped to measure, this research could lead to a quick test for E. coli that third world countries could use on site perhaps, instead of having to culture samples overnight in a lab and using expensive equipment. Instead, they would just take a little device out to the water, quickly test for E. coli in about 10 minutes, and know if it's there.